next speaker is also from Seattle Children's, Guillermo Romano, and he'll talk about efficient targeted gene modification primary human hematopoietic cells using co-delivery of nuclease mRNA and AAV donors.
Thank you. This is the one we're using. Great. So my name is Guillermo Romano-Ibarra, and as mentioned, I'm a scientist in Seattle Children's Program for Cell and Gene Therapy. And I'm talking about today is one of the big challenges in cell therapy, which is the targeted delivery of genes and their use in autologous therapies. So the aim of the project that I'm talking about today has been to develop a high-efficiency approach for gene modification. Importantly, we're going to be using primary cells here, so this is a little bit more clinically relevant. And so I'm going to begin by reintroducing a novel CCR5 nuclease that we've created in collaboration with Bluebird Bio. I'm going to present how we're using this nuclease in a co-delivery approach that allows us to achieve 60% targeted gene modification in primary T cells. And then I'm going to show you how we're using this approach to target clinically relevant gene cassettes into CCR5. And I'm going to present one example with relevance to HIV, and then my colleague will follow up on me and show some functional studies where we've knocked in chimeric antigen receptors into the CCR5 locus. So as has been covered pretty extensively today, CCR5 is an important co-receptor involved in HIV entry. And our group has a history of engineering homing endonucleases. So what we've done was assembled a homing endonuclease to a TAL binding domain. And this is what we call a megaTAL, and this gives us a monomeric cutting platform with binding-dependent activity. And this allows us to achieve 80 to 90% CCR5 disruption in primary human T cells. And so this was the subject of an earlier talk today by Bish Paul, where we were challenging cells and using this nuclease to protect against HIV. For the scope of this talk, what we're trying to do is not only disrupt CCR5, but subsequently reintroduce new genes that can have therapeutic benefit in the context of HIV, but as well as other using CCR5 as a safe locus for other clinical challenges. So, as has been covered pretty extensively by Dr. Porteous, Paula Cannon, and Dr. Scherenberg, the way that we're going to deliver our nuclease is through mRNA transfection. This gives us rapid, high-level, and importantly, transient levels of our nuclease that are expressed. And then we're going to follow this up with donor template delivery through AAV. And so this has also been explored today and has been shown notably by Dr. Porteous that you can achieve homology-directed repair or recombination with and without double-stranded breaks. And so this is what we're moving forward in our approach. So what we've created in our lab is an AAV donor template. And in this case, it's a GFP gene cassette driven off an MND promoter, and it's flanked by regions of homology to the CCR5 gene that flank the cut site of our nuclease. And so we hypothesized that in the presence of a double-stranded break, if we deliver our construct, we should see recombination of this gene cassette, and we should see sustained expression over time that will outlast the diluted episomal AAV. In contrast, to control for that, we have another viral construct, which is similarly a reporter gene driven by the same promoter. In this case, it's BFP. And we're missing our homology arm. So even in the presence of a double-stranded break, we should see limited sustained expression of this gene cassette. So the way we're going to do this, again, one of the emphasis has been clinical relevance. So we're going to start from cryopreserved cells, primary cells that we've processed in our lab. Immediately after thawing them, we're going to stimulate them on CD3, CD28 beats for 48 hours. We allow the cells to rest for 16 hours. And then what we call time zero is when we actually electroporate the cells with either megaTAL, so that's our nuclease, or a control mRNA as a transfection, just to measure our transfection efficiencies. We incubate the cells at 30 degrees, as was explained by Dr. Scherenberg, and then we're going to monitor the cells for 2 weeks using flow cytometry. So when we do this, we get great results, and this is what we see. So to orient you, I'm going to show GFP on the x-axis, BFP on the y-axis, and we see that with BFP mRNA transfection, we see a transient transient level of our gene cassette or of our protein, which is not sustained over the 2-week period. Similarly, when we deliver our experimental construct, so this is the gene cassette with homology arms, we see a high level, about 60% expression on day 2, which is lost by day 16. Now, we hypothesized again that in the presence of a double-stranded break, we should see sustained expression of this gene cassette, and that's what we see. We see that we go from about an 80% transient expression down to about a stable 60% gene expression by flow cytometry. By comparison, when we look at our control AAV, so this is in the absence of homology arms, we again see a high level of transient expression, but even in the presence of megaTAL, we see about a 10-fold lower expression by flow cytometry relative to our experimental HDR template. This is highly reproducible. We're looking at a summary of 25 different experiments across 14 healthy donors. We have an average of 60% gene modification with low variability, and importantly, especially for the use of this approach in CAR T cells, we have identical results in CD3 bulk populations as well as isolated CD4 and CD8 populations. So it looks great by flow cytometry. We want to validate this molecularly. So we're going to use primers that flank and that sit outside of the homology arm, so you won't amplify any episomal AAV in the cell. And what we're looking for is we're trying to amplify the product of HDR, in which case gives us a 4.4 kilobase product. When we do this, we can only amplify that product when we co-deliver our repair template with our nuclease. Next, this is especially important in the context of HIV. We want to be able to modify both alleles. So single modification of CCR5 will not protect the cells against HIV. And so what we're going to do here is try to quantify the degree to which we're achieving biallelic modification. And in this case, we're looking not only for biallelic disruption of CCR5, but our ability to deliver 2 copies into each cell, so 2 copies of GFP into each CCR5 gene. So we're going to single-cell sort our cells following a regular experimental protocol, and what we can see is that as we increase the amount of donor template that we make available for our cells as in the presence of a double-stranded break, we can increase the amount of biallelic modification. And our approach at our highest doses of AAV allow us to modify 82% of the cells biallelically. So 82% of our GFP-positive cells have biallelic modification. We also want to test the stability of our product in vivo. So we're looking at, we transplanted the cells after a 10-day expansion into NSG mice. We see that when we harvest spleen and bone marrow after 4 weeks, we see equivalent engraftment across all 4 groups, so untreated cells, megaTAL treated cells, and then our dual delivery approach. So we were curious to see whether CCR5 would affect engraftment. In our hands, it does not, or the CCR5 disruption. And what we're looking at here is we're seeing that in our dual delivery approach, we can retain the amount of GFP expression that we get from these modified cells. Furthermore, we can validate the fact that this GFP expression correlates with our predicted HDR PCR product. We also wanted to compare whether or not this nuclease with this co-delivery approach is translatable across a number of different nucleases. And so what we did was we developed talons to the CCR5 locus, and so we're cutting at a nearly identical site as to our megaTAL. There's a number of different ways that you can assess NHEJ, but in our hands we can see equivalent disruption rates of CCR5 using both of these nuclease platforms. And we can achieve a high level of gene modification using TALEN. So we're looking at about 50% gene modification at our highest doses of AAV. Curiously, when we compare head-to-head to the megaTAL across a number of different AAV doses, we can see a higher level of gene modification as seen by HDR using our megaTAL nuclease. So moving forward into clinical applications, we're going to focus on utilizing the megaTAL nuclease. And the first thing that we wanted to test was see if whether or not we could move from T cells and apply this technology into stem cells. So the most clinically relevant source of stem cells would be using adult-mobilized CD34 cells. And so we have a slightly modified protocol, and we have a great group of people in our lab currently working to make this even better than what I'm going to be presenting right now. And so we can see that we have a high level of BFP expression. This means that we can transfect our CD34 cells. And when we use our co-delivery approach, we have a sustained expression by day 10 of about 20% gene modification. And so there's been a number of different groups, including Paula Cannon, that's presented data very similar to this, and so our approach is consistent with what other groups have presented. And we can validate the fact that this is happening through HDR by amplifying the correct PCR product. So another application, and this is where we're moving into the HIV, moving back into the T-cell model, we've created a donor template that codes for the C46 peptide, which was explained a little earlier today, but C46 is a fusion inhibitor that prevents the HIV envelope from binding to the T-cells. And so you could envision, as we're simultaneously knocking out CCR5, one of the concerns that we mentioned earlier was whether or not we would be selecting for CXCR4 tropic viruses. By delivering a C46 peptide, we would be protecting against all tropisms of the virus. And so in our construct, as Dr. Porteous alluded to earlier, sometimes T2A peptides can be a little misleading. So what I'm showing here is that our GFP expression correlates with surface staining of the C46 peptide as measured by the 2F5 antibody. And when we subject this construct to our experimental protocol, we can see that we have a sustained level of about 30% of our C46 construct in T cells. So a technical summary of this work, we're looking at the co-delivery approach that we've developed, allows us to achieve 60% gene modification in primary T cells, and we're looking at about 20% gene modification in adult stem cells. We've been able to this method is compatible across a number of different nucleases. So I've showed work with the megaTAL as well as TALEN. Dr. Scherenberg presented TALEN work at a different locus using the same method. And importantly, we've been able to deliver therapeutically relevant genes into CCR5. And so what I showed you guys the C46 peptide. In a second, Malika Hale is going to come up here and tell you how we're validating the 2 different CARs that we've knocked into the CCR5 locus. And for our community viewers, what I'd like to emphasize is the fact that we've developed an approach that would allow us to take patient T cells and then engineer them so that they're resistant to HIV. And subsequently, beyond protecting them against HIV, we can introduce various genes that would allow us to modify the behavior of these cells. And so the modification of these cells using the approach that I've presented today is relevant to a number of different clinical applications, not only for HIV, but as well as certain other cancer applications or immune deficiency applications. I'd like to stress that this has been a huge undertaking by a lot of people in our group, very collaborative, and no one person can take credit for this, and really speaks to the leadership in our program for cell and gene therapy, led by David Rawlings and Andrew Scherenberg, and I'd like to thank them for their leadership. And with that, I will take any questions, and hopefully I will know what the answer is.
Any questions? Go ahead.
I was going to ask on the C46 again.
Sure.
I'm getting tired of it. But the C46, that's expressed in the surface, so there's a potential to be a T-cell epitope, has the potential to be a B-cell epitope. And so why won't there be sort of selective clearance of the C46 expressed cell?
So could you say that one more time?
So as I understand it, the C46 is an improvertide-like peptide that's expressed on the cell surface. Is that correct?
Yes.
So that raises the possibility that it could serve as a T-cell epitope, meaning CTLs, for example, could clear cells expressing that. Also raises the possibility that it would be a B-cell epitope because B-cells would recognize it on the surface. So wouldn't that give it a disadvantage?
You know, I don't know what the answer to that is, honestly. You're digging into the deeper immunology here. I'm not sure if David would have any hypothesis there, but.
So in any time in all of our studies, we've not seen it, but we've done this with delivered via lentiviral vectors, but in these studies we've actually done conditioning, so the animals or patients will receive then some conditioning. So exactly, so they might be tolerized, and in the previous studies now by the German group, I think they didn't see, they infused the cells, but unfortunately they didn't persist long enough, but they didn't really see an immune response, but this could have been because the cells just disappeared without the immune response.
Hi, you showed data where you were looking at the level of biallelic
Sure.
knocking of your
Mhm.
CAR as a way to make sure you got rid of CCR5. Because you can also get rid of CCR5 and monoallelic knocking if you have an indel as impressive.
Sure. Yep.
Did you also look at that?
So we didn't. And so what I was looking at was measure the, and I say I very loosely, we were looking at the biallelic delivery of the, and it was only for the GFP cassette. So we haven't done biallelic assessment of our CAR. But for our GFP cassette, we're only measuring, we're basically PCRing on a single cell-by-cell basis, and you'll get a hit. And so you'll either see wild type or NHEJ band, or you'll see the increased size that corresponds to the HDR product. And so that's all we were quantifying. So certainly there are probably events that we're measuring as non-biallelic, which are monoallelic HDR and probably monoallelic disruption. And so we haven't quantified that. And so, oh, you know? Okay.
By side, we could really research a little bit, but by side in the monoallelic GFP positive cells, we got like 24, 25% NHDA a couple times.
So I also had a question there. Was there the MOI you used for the AAV delivery was pretty high?
Pretty
Is there
high.
any toxicity?
No, so well, so there's toxicity associated, right? 140,000 MOI, and so you see some toxicity. Now the AAV by itself, you don't see toxicity, and when you do a mRNA transfection by itself, you don't see toxicity. As you start combining these approaches, you see some level of toxicity, but nothing that's not recoverable. So I think it's just inherent to the manipulation of the cells. And so any one individual part, everything stays above 80% viable early on, like day 2. When you measure our viability in cells that are achieving HDR, they might drop down to high 60s, low 70s, but it's all within the range of what we consider to be acceptable levels of toxicity and modification.
Great. I think we should move on
Cool.
to the
Great.
next speaker from children. Thank you. Next, anti-CD19 CAR to the CCR5 locus in primary human T cells.
Yeah, I do have a pocket. I do not. Thank you. This thing says avoid exposure in really big capital letters, so I'll try not to expose anybody. As has been subtly hinted for a little while, I'm going to be talking about integrating chimeric antigen receptors at CCR5 using technology developed within our program at the Program for Cell and Gene Therapies at Seattle Children's. And I put my community slide first because I thought this was kind of a cool way to do it. I'm a simple person, and this is how I think about it too. So the idea is that we're accomplishing two things in one. We have the targeted delivery of therapeutic genes. This is done traditionally and successfully for a lot of gene therapies with lentiviruses and in some cases retroviruses. And then we're simultaneously, hopefully, protecting these cells from HIV infection. And then thinking about how we can use this clinically, one, I think, potentially interesting use of this is to deliver chimeric antigen receptors to T cells. And then you can imagine that this might create chimeric antigen receptor cells that can be used in HIV patients, and they could be reprogrammed to attack cancer, as is being currently used, or as Thor talked about earlier, HIV. And then Guillermo just covered all this. We have this nice method that's working well with us with delivery of a nuclease with mRNA and then a donor template with AAV. It's working well for us, I should say, and a number of other groups today. So you can imagine a bunch of different things that we could use. Guillermo showed the C46 fusion inhibitor. You could maybe imagine some sort of like immunomodulatory factors that you could use. You could imagine putting something in that has direct anti-HIV activity, which we will get to. But first I'm going to talk about something that you could maybe be used to treat other pathologies, possibly in the context of HIV-infected people. So one of the best validated, I think, chimeric antigen receptors that has a really nice so far history in clinical trials is the CD19 CAR. So we thought we'd start with that. It's not completely trivial because HIV-positive people do, over time, tend to develop B-cell lymphomas at a higher rate than the general population, so this might, in fact, be a population that could benefit from having access to the CARs. They're traditionally delivered by lentivirus, which would leave these cells, in theory, in HIV-positive people to infection. HIV-positive people have been so far excluded from these trials for that reason, and I assume some others that people can probably think of. But doing it this way might create a more predictable, consistent product that would be protected in the patient. So this is the construct that I'm using. It looks a lot like the construct that Guillermo just showed. We have these flanking homology arms, and then I stuck in the successful UPenn CD19 CAR that's probably familiar with people, and then I slapped on a BFP because it's easy to see. This is just the same protocol. We deliver the nuclease, we deliver the AAV, and then we read out, and then you can enrich for BFP. And then in those enriched cells, you can PCR, doing the same PCR across the locus, and see a band that's consistent with seamless integration of just our CAR construct right at CCR5. And then if you look at these same enriched cells and actually look on the surface, staining with protein L, you can see that we are, in fact, getting detectable CAR on the surface by this stain in these enriched cells. And then we'd like to test efficacy, obviously, and we'd like to compare to what we think is probably the gold standard, which is lentivirus. So we just slapped this same construct into a nice lentiviral backbone. Lentivirals are banger at expressing things, so you can see the MFI is higher. And so these are, for experiments where I'm comparing, I've sorted on consistent, so we have the same percentage of cells that are positive, but they're not super well controlled for expression. So I think in a lot of the steps, the lentivirus is expressing more. So this is a system that I'm using to test in vitro. It's pretty simple. 2 different target cells. One of them overexpresses CD19 and GFP. The other overexpresses BCMA, which here is just a totally irrelevant B-cell protein that the CAR should not care about, on a different fluorophore. And then by adding the CAR to either of these, we can look for activation and degranulation and see that those responses are specific only to the CD19 positive cells. And then because they're different colors, we can mix them together in the same well and look at specific killing. So showing first activation by CD137, I'm showing just an unmodified T cell mixed with CD19 positive cells, not a lot happening. The targeted CAR and the lentiviral CAR both appear to me to be expressing upregulated CD137 at this time point. And I kept getting in trouble trying to show this when I was showing this to my group. I wanted to call these equivalent. They're not. In these assays, the targeted CAR is responding on average, which much stronger at this time point. And then this is degranulation by CD107, so same thing. This is a specific response. You only see it with the CD19 positive cells. Nobody gets it with the BCMA, and it's within 10 percentage points of the lentiviral CAR, and if not anything, it's higher here with this assay. And then that's just showing indications that the cells are getting sufficiently jazzed. To look at actual killing, we do this, in theory, 50-50 mix. These are our irrelevant decoy cells, and this is the CD19 positive cells. If we can do the whole targeting process, and then in the part where we enrich, we sort on the negative cells and then add those. So these had all the same experience, except they happened not to have gotten targeted. Adding them to culture doesn't do anything, but then when you add the CAR cells that were sorted on BFP positive, you basically lose this whole population. And this is replicable across a couple donors and a number of different experiments. So that's the CD19 positive CAR. We wanted to just show briefly in one of these little experiments that everybody hates that they can survive in something that's fuzzy and happy and moving around. So we injected T cells, looked out 5 weeks later. The marking what was in was pretty low, so we weren't sure if we would see anything. Those are the human cells, and then if you look within those human cells, we get about equivalent marking going out. So this gives us some positive data to go into future in vivo actual killing experiments, looking at actual, hopefully, death of target cells in vivo. So that's one CAR, one target. There's been another fairly successful CAR, which is this anti-BCMA CAR that's in trials at the NCI right now. Again, this is not totally irrelevant. Plasma cell disorders, there have been some reports that they end up being more common as HIV-positive patients age. So we have this CAR, same thing, slapped it in And this time we're using the same assay, but it's just split the other way around. So the CD19 positive, these guys are the decoys, and now the BCMA IRF1s are the targets. So looking quickly at activation with those, we see that these cells are not super activated on their own. When you add the CD19 positive targets, they don't care that much. They do react with the BCMA positive targets in a way that the CD19 CAR does not, and this is the same thing that you see with the degranulation. So finally, for HIV stuff, so Thor showed some of our experimental HIV CARs. Here, I'm just sort of plugging them into this system that's worked nicely for us. We have a cell line where we have the uninfected parental cells are M-cherry positive, the infected HIV positive cells are GFP positive, and so we can look again using the same sort of format. So I'm showing activation here by CD137, so I have the CD19 CAR as a control. Not a lot happening with the HIV negative targets in this assay. This is targeted CARs from 3 donors. And then when you add the HIV positive cells, I think we can argue that we do see a response there. So that is our protected CAR activation, killing. I just have a little bit of data to show. So this is the HIV negative cells are in a box here. I didn't mix them in this one just because this was one of the first ones that I did, but here's they don't seem to care whether it's a CD19 CAR or an HIV CAR. However, when you have the HIV positive cells adding the CD19 CAR, you have about 24% of the cells that are in that well are GFP positive. We couldn't see any when we added the HIV CAR in this experiment. This is 48 hours after addition. So I think hopefully I've at least convinced myself that we can get efficient gene targeting of the primary cells, that they activate, that they can kill, that it's at least comparable to lentiviral delivery, and that they'll survive in a mouse. I think this is 3 possible therapeutic extensions. I think people can probably think of more. There's these malignancies that HIV positive people are at increased risk for, and then of course there's finding means to target the latent reservoir itself using this technique. And this is a huge program of a bunch of awesome people. Thank you so much.
Thank you, and thanks for keeping it succinct. So any more questions? Since we're a little bit, oh, there's one. Good.
We haven't really looked. I've done like 12 cells of single cell PCR. So, in that case, it's 60% biallelic, but I wouldn't put that on a slide. I think that's definitely worth looking at, and it definitely seems to matter more for this application.
Are you doing, so is your single cell PCR at that full base?
No, we're just doing flow single cell sorting into plates and then doing PCR from that. It's been pretty well worked out by people in our lab who are on the B cell side who do that often, so we have good experience with that.
Okay, thank you very much again. One more presentation on CAR T cells, now from UCLA by Masakazu Kamata.
Perfect.
Hopefully close. Thank you.
Thank you. To use this keyboard? Okay. All right. So I'm last speaker in this section. Just want to skip this one and just want to start with summary for the community. So the key question which I was working is how to design the CAR and what type of the T cell want to use for the CAR carrier vehicle. And want to skip this too. And what we should do now for the future therapy is how we can maintain those CAR modified T cell in the patient for a long period. So this is a really old CAR which developed almost 20 years ago. Many people hate since it's failed in a clinical trial, but still having a big advantage compared with the current single chain based cross therapy. So CD4 data having the extra domain of the CD4 and then transmembrane domain and with CD3 data chain. So it's called the first generation CAR. The 3 clinical trials have been done, and then what we've learned from those clinical trials, the vector is safe in the T cell and then stable over 10 years, but the constitution level is so low, and efficacy is almost nothing or it's very minimum. So how we can overcome those problems? These are my hypotheses here. So first of all, CD4 data is having the full length of the CD4. That's why it supports virus infection. So we're going to protect the virus infection to the CAR-modified T cell. Then second of all, it's more general problem with using the gene-modified T cells. Before we transplant to the patient, those T cells expand ex vivo extensively. That means that those T cells may be in the end of stage, so they're going to die off very quickly once stable in the patient. Lastly, the first generation of the CD4 data CAR is not having any costimulatory domain. So first of all, I just tested whether those CD4 data can support HIV infection into the CD8 T cell. I saw the same thing with the CD4 T cell. But I just want to point out, when CD4 T cells, CD8 T cells are modified by EGFP, of course, they are no infection, but by modifying with the CD4 data, they are infectable with 3 types of different viruses. So how to protect the virus infection to the CD8 T cells, since we are developing the 2 different shRNA against HIV. First one is the SH1005. It's targeting CCR5 developed by Dr. Johnson and then Dr. David Baltimore. It's around 8 years ago. Then second is shRNA, which can block the HIV1 transcript itself. So by having both 2 shRNA, we can protect CCR5 tropic HIV as well as the CXCR for tropic HIV. Then we combine those 2 shRNA together with the CD4 data expression CAR. So one vector can protect from the HIV-1 infection as well as it can kill the target, in this case, HIV-1 infected cell. So this is just simple test whether those CD4 data with 2 shRNA can induce HIV-1 specific effector function by cytokine production as well as killing. So compare with the CD4 data CAR, triple CD4 data CAR, now I call the triple CD4 data, can kill the HIV infected cell, same as the CD4 data function. So whether those triple CD4 data CAR can protect CD8 T cell from the HIV infection, as you expect, of course, it's broad compared with the CD4 data. And this is by alpha tropic virus, but I tested with NL43. You can see the similar result. Then this is the last slide of this section is CD4 data. Again, it supports HIV infection. That's why, especially by infecting with NL43, those cells are killed off almost 50%. But by using a triple combination, we can protect those cells from the cell death induced by HIV infection. So this looks good, but always it works on bench. But once we transfer to the clinical or in vivo, it's hard to get a good result. So I tested those vectors using a BLT system. I won't talk too much detail, but the week 0 after transplant, the CAR-T modified T cells and those virus copies dropped very quickly, stayed for 2 weeks, but it's going up. This is the most important issue is using the current generation, our generation of the CD4 data, since it doesn't have any costimulatory domain. Also, the T cell, which we use, extensively expanded at ex vivo. So those are almost terminally differentiated. So how we can overcome those 2 problems? So first of all, how to minimize those ex vivo expansion, or how to maintain growth ability of the T cell even after transplant to the patient. Also, what kind of the cross-stimulated domain may help to induce the memory function. So this is a very busy cartoon, but I'm focusing on using the T stem cell. Many people, I think, knows about this population can be a latent reservoir for HIV infected patients. So the T cell differentiation starts from T naive and then get to the T stem cell, and then further differentiate to the having the more potent effector function. But what T stem cell is, that one having self-renewal ability also can differentiate into the central memory, effector memory. So ideally, by modifying this population, you can keep maintaining the CAR T cell in the patient, ideally forever. But so far, what we know is that those cells are stayed over 14 years in the patient. So what I did is I modified T cell, CD8 T cell, in this case, with CAR CD4 zeta, triple CD4 zeta, and then tested whether T stem cells modified with triple CD4 zeta can maintain the effector function as the same as the effector's memory T cell. So I could see the difference. But both having a good HIV specific effector function. Then I won't explain too much about this system, but since BLT system is taking over 6 to 8 months to see the result, so I shifted the in vivo system to use xenograft model. So basically, the transplant, the Chagas cell, which constitutively expressing HIV envelope, then right side having the jacket, and the left side having the BJAB also expressing HIV1 envelope, then whether those CAR T cell can kill those tumors. That's the experiment which I want to show here. So this is a control CAR. I'm using a CD19 CAR as a control. You won't see any killing, so you can see the tumor growth on both. But by using the triple CD4 zeta CAR, modified T stem cell, you can kill both tumor. The last slide, this is the last slide, and what kind of the co-stimulated domain we should use? It depends. Well, I can tell you it depends. I'm testing the 4 different co-stimulated domains, ICOS, 4-1BB, OX40, CD20. In case of a CD19, which I tested, all these having the same level of the effector function. However, by using a CD4 zeta, it's having a difference. Difference means its specific killing will be different. Also, one issue is using a CD28 together with CD4, it's inducing the more differentiation. So you cannot maintain the T stem cell. So this is a conclusion slide. Now we could make better CD4 zeta CAR vector, and then those vectors can modify in T stem cell, and then additional co-stimulated domain can make better CD4 zeta now. I'm testing those co-stimulated domain added triple CD4 zeta CAR using the xenograft system now. So next time I'll try to present that data. So this is the last slide, and I acknowledge to everyone working with me, many people from the Channel Lab, also Dr. Otto Yang helps killing us there, and then Dr. Scott Kitchen helps BIT experiment, and Jay is giving me a good discussion. Thank you for attention.
Questions? So the BLT data is very interesting. The viral loads seem to come back up. Have you sequenced the virus? Did this change, or did you just lose the T cells?
I didn't do that. After they came back, it's after 6 weeks, we checked the CAR T cell in the BLT, but I couldn't detect it. So T cells are not stayed in the BLT. two reasons. Maybe since the genetic signaling maybe induced the T cell more effector level. So that's why the T cell just disappear in that system. We better to test in the monkey using the autologous systems.
All right.
Thank you.
Very much.